that the sections of the book that are listed as going along with the lectures that we're doing, but the focus of the exam problems is going to be in the lectures. So today I'd like to talk about nucleic acid chemistry, a few different things that can happen to nucleic acids, to the bases in those nucleic acids, and also to the backbone of DNA or RNA. And that's going to lead into some discussions of some of the technology that we use in the study of nucleic acids, most importantly, how we can sequence nucleic acids and how we can synthesize them. And then finally, we're going to discuss briefly the fact that chromosomes, the actual full complement of genetic material in a living organism, is a very, very large single molecule, essentially, and that causes some substantial problems for the cell that we're going to be discussing in future lectures. But the first thing I'd like to talk about is this idea of melting and hybridization. So because DNA is a double helix, and RNA can form a double helix as well, and it has a perfectly complementary strand. But as you heat up nucleic acids, they can start to separate. And that separation can be partial, and that's sort of what's shown in this electron micrograph over here. The regions of DNA like here, those are double-stranded, and these little bubbles are regions where it's partly melted. And so this denaturation and annealing happens as you heat the DNA, and eventually, particularly with shorter pieces of DNA, the DNA can separate entirely. And once that happens, the DNA molecules basically need to find each other before they come back together. So this process can move back and forth quickly. You can imagine as the temperature changes, these bubbles opening and then closing again. But if you completely melted these two strands apart, they would need to find each other before they came together again. So, one question might be, why is it that some parts of this DNA have started to melt apart whereas others haven't? And one of the major factors that controls the melting temperature of DNA is its GC content. So basically, regions of DNA that have more G's and C's have a higher melting temperature, so they form a more stable double helix than those with high AT content. And certainly one part of that is the fact that the GC base pair has 3 hydrogen bonds, whereas the AT base pair has 2. And it's actually slightly more complicated in that there are contributions from the stacking interactions that I think actually aren't well understood even now. Empirically, we know that when we have G's and C's next to each other, they stack in a particularly favorable way, and when we have lots of T's and A's next to each other, they stack in unfavorable ways. So that certainly contributes as well. But basically, you can think of high GC content is more stable. Then the other point is that the DNA and RNA double helices don't have the same melting temperatures. That actually the DNA pairing is the least stable. It has the lowest melting temperature. It's easiest to separate the strands. And then RNA-DNA hybrids are intermediate, and these RNA-RNA duplexes are actually the most stable and have the highest melting temperature. And so you remember that these actually, that RNA-RNA double helixes actually take on a different physical form, this A form, which is shorter and wider than the DNA. And correspondingly, they have at least somewhat different physical properties. So when, yeah? Yeah, we'll talk about that in a minute. So this is, so the question was the fact that RNA is chemically less stable in the sense that you can actually break the backbone of RNA spontaneously in a way that doesn't happen for DNA. That's actually something that we're going to discuss later in this lecture. So I should say that this that we're talking about here is not the chemical stability of a single strand of RNA or DNA, where DNA is dramatically more stable than RNA. It's the base pairing of RNA with RNA is tighter, has a higher melting temperature than DNA-DNA base pairing. Yeah, do you have a... That's a good, so the question is how uracil content affects that, and the answer is very little, and we'll actually discuss in a minute how the difference between uracil and thymine is actually not relevant for base pairing. It actually has a different significance for DNA. So I mentioned the fact that you could melt apart strands of DNA. In fact, you melt them apart entirely in solution, and then when you cool them back down, they can find each other by base pairing and then sort of zip together. And if you have a situation where you've actually mixed two different DNA samples, they can actually form what are called heteroduplexes. So this is if you have some nucleic acid strands in the blue sample that are complementary to some in the red sample. When you melt them both apart, mix them together while they're still melted, and then cool them back down, that they can find complementary strands, whether they came from the same sample or a different sample. So, for instance, this was human DNA, and this was mouse DNA, which is actually some regions of pretty high complementarity, some regions where the bases in the genome sequence are almost the same, that you would get many regions where a human sequence actually encountered a complementary mouse sequence before it encountered the complementary human sequence, and those would stick together. And likewise, you could do that for a sample of RNA that you might have and a sample of genomic DNA, and then that RNA might find the genomic DNA that it came from by hybridization of those complementary sequences. And complementarity doesn't have to be perfect to get this matching. There can be some amount of mismatches. The more mismatches there are, the lower the melting temperature. But you can still detect regions that have high similarity, but not perfect identity. Yeah. The question is, are functional significance in heteroduplexes? So we use it technically quite a lot in a number of experiments. When you're going to find a sequence of DNA or a sequence of RNA, we can create the sequence that we want to look for, and then in different ways we can actually probe the sample that we have, DNA or RNA, to find things that are complementary. And that's actually sort of the idea here. So in the previous slide, I showed you that you can have two solutions of two different DNA samples, for instance, mix them together and they can find each other. Actually, a related thing that you can do is you can have a solution of DNA, and then you can also have a different DNA sample that you physically immobilize on a membrane that you can bind to DNA. And actually, when you take that membrane with a single strand of DNA, you dip it into the solution, the DNA in solution will find the complementary DNA on the filter, stick to it, and you can detect it that way. So the idea is that, for instance, if you had DNA in solution that was radio-labeled, and you had a variety of different DNAs on this filter, and you wanted to know which of your blue samples on that filter was complementary to your radio-labeled probe, you could mix them together, and the radio-labeled probe in solution would stick on the filter only to the DNA that's complementary. And that way, you would have the labeled probe DNA sticking only to the place on the filter that had complementary DNA. And so this is a technique that's used quite widely in molecular biology to detect specific genes either in DNA or in RNA. So essentially, I mean, this is the chemistry that happens essentially between two nucleic acid strands. They find each other based on complementarity, and they pair together. And I guess the next thing I'd like to talk about is chemical changes that can happen to individual bases in DNA or RNA. And to remind you, of course, the information content of DNA is the sequence of bases. And in almost all cases that we're going to talk about, that information is copied and read out by the cell by using complementary base pairing. So any sort of changes to those bases that disrupt the way that it pairs will cause changes in the information in DNA. And this, if this is happening in the DNA, that effectively causes a mutation. So one of the most prevalent kinds of chemical change that can happen to bases is deamination. So so spontaneous cytosine deamination happens in some sense relatively slowly. So the rate of this reaction is about once per 50,000 years for a single cytosine. But actually your genome has about 3 billion cytosines in it, in each cell for a diploid genome, which means that in the course of this lecture, each cell will experience about 5 spontaneous cytosine deamination events. So the mechanism of cytosine deamination is shown here. To remind you, all of the bases have a tautomeric form. This is for cytosine. This is the amino form, so almost all of the time this is the form that cytosine exists in, but occasionally it can undergo this immunotautomerization. And in that form, this carbon is subject to nucleophilic attack by water, which adds here and can then rearrange by the loss of water into uracil. So this is the mechanism of this cytosine deamination. And the effect of this on the cell is that if you had a GC base pair, that GC base pair would at some very slow rate become a GU base pair. And then after DNA replication, of course, the G would still pair with the C, and so one daughter cell would get an unmutated copy of this gene, but this U would actually prefer to pair with A. And so in fact, an A is inserted there. And so effectively, after this spontaneous CDU change in one round of DNA replication, you've introduced a mutation where a GC base pair, the genome became an AU base pair in one of the daughters. So this is, of course, a major problem for the cell. And I think to get back to a question someone asked earlier, this is the reason most likely that thymine and not uracil is present in DNA. That the methyl group on thymine distinguishes it from uracil. And in fact, there's a specialized repair system that detects uracil in DNA, which arises only as a result of this kind of mistake, and corrects it back to cytosine. We'll discuss the exact details of how that happens later. But the uracil in DNA is very efficiently removed and repaired back to a cytosine. However, in ancient DNA, so this is DNA in an organism that has died many tens of thousands of years ago. Of course, this repair process no longer happens. And when people first started sequencing DNA out of ancient bone samples, and here showing a picture of the people who sequenced the Neanderthal genome out of Neanderthal bone samples, but people saw the same thing in ancient DNA from mammoths and so forth. So there is a very substantial rate of this cytosine deamination that happens. And in fact, about 50,000 years, which is the rate of this cytosine deamination, is roughly the time scale along which people can actually successfully sequence ancient DNA. And even when they do see this high frequency of CDU changes. So this cytosine deamination reaction I mentioned happens spontaneously in living cells where it's corrected, and happens in isolated DNA where over the course of tens of thousands of years it will degrade. It is also something that the cell does deliberately in certain cases when it wants to introduce mutations that change the coding content of DNA or of RNA. And a few places where this happens most prominently are, first of all, in lymphocytes, which when they're developing in order to make high-efficiency antibodies, actually deliberately mutate the genes that encode those antibodies. And this is actually done by an enzymatic cytosine deamination. And there are also some cases where RNAs are edited in the same way. That there are APOBEC enzymes that in some cases change cytosines to uracils in an mRNA. Now, of course, mRNAs normally have uracil, and there aren't pathways to repair RNA sequences in any case. So when you change the cytosine to your uracil in an RNA, it actually changes the protein that RNA encodes. And in addition to the deliberate editing of cellular mRNAs, there's also targeted cytosine to uracil changes in viral RNAs that occur as an antiviral defense mechanism. So this is a reaction that can occur spontaneously, or it can be catalyzed by enzymes. There is another deamination that can occur on the other amine-containing base, adenine. And so this doesn't happen spontaneously at nearly the rate as cytosine deamination. And so it's not a sort of a major point where there's sort of a specialized DNA repair pathway that looks for adenine deamination, but it is something that can happen catalyzed by enzymes in a few specific cases. This is the adenosine deaminase that can change adenines in RNA, and it can happen as a result of exposure to certain chemical mutagens that make this reaction occur more quickly than it would in sort of the uncatalyzed way. And so when adenine is deaminated, it becomes hypoxanthine. So hypoxanthine is not a base that occurs naturally in DNA or RNA. And so it is, and so the name of the base alone is hypoxanthine. Inosine is the name of the nucleoside or the nucleotide. It's called inosine or inosine monophosphate. It's a bit of confusing nomenclature. But the important point here is that the deamination from adenine to hypoxanthine changes the pattern of hydrogen bond donors and acceptors on the base pairing region of adenine to hypoxanthine. And so what you can see is that actually the pattern here is more similar to the pattern on guanine, where in this position you have a hydrogen bond acceptor, like on guanine. In this position you have a hydrogen bond donor. And as a result, hypoxanthine or inosine will actually pair better with C than with U. And again, putting this inosine, hypoxanthine, in a nucleic acid effectively causes a mutation. And so, guanine, so if you could get out your eye clickers now. So here's a third case where you can have the deamination of a base. So guanine does also have an amine on it, and it is capable of being deaminated to xanthine, which is shown here. And sort of for your reference, I've put up the canonical AP and GC base pairs here. And so the question here is, which bases can pair with xanthine after this guanine has been deaminated into a xanthine? And so I'm told that last time you guys cannot see the timer on this. So I'm going to start the timer now, and I'll give you about 90 seconds to talk amongst yourselves about which base pairing you expect for xanthine.
Oh. This is guanine's turn to deaminated.
No. But it kind of looks like...
Yeah, it is.
Oh, right here.
Oh, that's weird.
That is weird.
That's not how it should work. Look at that one.
Okay, so it's been about a minute, so if you could wrap up with answering in the next 15, 20 seconds.
Going to Google.
Okay, I'm going to go ahead and close this now.
Is it C? A?
Okay.
Too late.
So, in fact, the changing guanine to xanthine doesn't actually change its pattern of base pairing. In fact, xanthine will still pair best with cytosine. So, if you look at these base pairs here, I just sort of drawn the change that happens in xanthine versus guanine. Of course, it does get rid of this hydrogen bonding interaction, but it doesn't disrupt these. And if we compare cytosine to thymine in this position, they're actually the same, so there's no reason that xanthine would prefer thymine. And I think the other principle that's important to remember is that in terms of sort of the proper size of the base pair in the double helix, it's important that we have a purine like adenine, guanine, or xanthine, and a pyrimidine like thymine, uracil, or cytosine, one on each side. If you have two purines, it's physically